in particular. And I'm going to share very specifically two abstracts that were recently submitted for meeting consideration, oral presentation consideration from work in my lab. And so I'm going to have you do a little bit of tweaking of those abstracts for the purposes of designing some oral presentation ideas. Okay, so I'm asking for your assistance after we've talked a little bit about some of the basics for storytelling. And I think if you even Google scientific storytelling, there's any number of things that come up. One of the things that I wanted to share just in this first slide is a little cartoon that is on a website, sarahelshafi.com. And if you have a chance to take a look at some of the aspects of this, I think there's some really interesting points that come to bear about how to put a story together to tell folks what it is that you do in the laboratory, what your research is all about. And I know that there are students on this workshop call that have been in the CPHAGES program. And one of the things that my colleague, Dr. Kenna, and I strive to do is to get students to get the experience of talking about the work that they do to different audiences. And today I want to have you think about a scientific audience versus a non-scientific audience. And we know that in each of those categories, there are obviously additional subcategories, but right now we're just going to focus scientific versus non-scientific for some of the discussion that we have today. Okay. So I have some storytelling workshop goals that I hope focus on today. First, I want you to think about why storytelling may have some value. What's the point? Why would we approach the work that we do from a storytelling point of view? I'll talk about some basic concepts in using a story. And this is based on a link that I asked Nandini to share with you, which is from a Nature article that was written by a PhD student, current PhD student at the University of Oxford in the UK. But this PhD student had a very interesting background, used to work essentially in the entertainment industry, and learned a fair amount of screenwriters' approaches to building a story essentially for a play or for a movie. And so some of that is going to come into play today. And there are ways of thinking about telling a story, which will include what's the compelling question that you're pursuing. You're the character in the story, and how do you make someone actually care about you as the character in the story? And what's the nature of the journey that you're taking as you talk about the work that you do? What's at stake? We certainly want people to think about what we're doing as important, as a contribution to either basic science or some aspect of applied science. But what is important about the work that we do? And then, certainly we want folks to have an interest to be persuaded that what we've been talking about is important, and what's the next act? What's coming next after we talk about the work that we're doing? So I want to discuss a little bit about how the story changes depending on the audience. And as I said, the CPHAGE students are familiar with changing up a story depending on the audience. You might have to explain what it is that you do to a middle school student. and you know the background that a middle school student has, and you might even have some sense of what excites a middle school student and how would you engage a middle school student compared to perhaps a grandparent or a parent who might have a greater investment in hearing about the work that you do, even if you were presenting it in sort of a dull or boring way, okay? Then I want us to break out into a few groups, and I'm going to share with you one of the abstracts in the first breakout session, an abstract that was submitted to the American Society for Biochemistry and Molecular Biology, and they're having a special meeting in October on the nucleolus, emerging roles of the nucleolus. And for those of you who have maybe heard of the nucleolus, but maybe can't remember what the nucleolus is all about, it's a membrane-less organelle within the nucleus. And that's actually where ribosomes are made. And, you know, hopefully folks have an appreciation for what ribosomes do. They are the protein synthetic machinery in the cell, and they get put together essentially in the nucleolus. And so there's a special meeting that's happening in October, and we submitted an abstract to that meeting, and I am invited to give an oral presentation based on the abstract. So we'll look at that, and I have some ideas that I'd like you to think about when you look at that abstract, and we'll talk about that. The second breakout session, hopefully we'll have time for that. Again, I want you to transform an abstract. This abstract is based on phage work that goes on in my laboratory, and this particular abstract was presented at a meeting by one of the graduate students in the lab. He gave an oral presentation based on this abstract, but he's planning to give another presentation in a month or so at another meeting. And so these are, I'll call them live abstracts from my lab on work that will be presented in oral presentations. And I want us to then talk about these, the ideas that you come up with. And then finally, I have a just for fun thing that hopefully we will be able to think about as well. So we'll get started with some basic ideas for an oral presentation. And this, again, is taken from the graduate student, Josh Ettinger, who is a PhD student, but he used to work essentially in the Hollywood realm. And the idea that you want to be able to engage your audience with a story, I think is a compelling one. How do we make that story that we want to tell compelling? And so he suggests that you could open your presentation with a question or something that hooks the audience in a way that will have them following the rest of the story for the rest of the time that the person is in fact engaged. So, you know, start with a compelling hook, locking the audience in from the start. And I think that's an interesting and important thing to try to do, because if you can if you get your audience hooked initially, hopefully they will stay with you through the rest of the presentation. Whereas if you get to the exciting hook, let's say a third or halfway into the presentation, you may have actually lost some folks initially. But you can also read your audience to some degree to get an idea of what it is that you're saying that may have been really the hook for their engagement, and to get them, maybe that's a way even for the next time that you do that presentation, in reading your audience, you would be able to see what worked and what did not. Then the question is, you know, it's all about the journey, according to Ettinger. You know, the research process itself is perfect for a story. You know, there are plots and twists, and the idea that you want your audience to appreciate the journey that you're taking through this research problem is important. And he suggests that you should invite the audience to understand the ups and downs of your research. And I think all of you have had some experience with things working in the lab, things not working in the lab. And sometimes we have a tendency to ignore the things that did not work, even though sometimes it's informative for the people that are listening to understand what didn't work. Not that you dropped your test tube on the floor, that certainly would have something fail. But we have a tendency not to talk about those things that were a challenge, let's say, in the research. But I think that when you tell a story, if you can convince your audience that you're excited about the work that's happening, what got you excited? What did you do that maybe was not such so informative? So these are personal experiences that sometimes can contribute to the story that you're telling. And then, of course, you have to explain what's at stake. Why would your audience care? And I want folks, when you're thinking of putting a presentation together, you've got to think about that question. Why does someone, other than just being polite, care about what I am saying, right? And so I think all of you have probably been in a circumstance where you started a story about the work that you're doing, and maybe even before you can get to the end of the story, someone has said to you, well, will that cure cancer? Well, how does that help with some biomedically related something or another? I mean, am I right about that being sort of the thing that connects people to research in biomedicine? They really want to understand, how is this going to help to advance some cure or what have you? So it's not always a cure that you're working on, but the basic science sometimes that you are engaged in is really important to help folks understand. And we certainly can appreciate what has happened, let's say, with the COVID vaccine. A number of people have been just startled about the speed with which those vaccines were produced. But how long have we known about messenger RNA? A long time. How long have folks been working with messenger RNA? A long time. How long has it been since you can, in fact, isolate DNA from a virus, sequence it in less than an hour, and then those kinds of bioinformatics tools that you can use to find out all kinds of information. So the general public doesn't know that, but part of our goal when we are talking about the work that we do is to get people to understand, even when we're doing basic work, how important that is to down the pipeline of where that might take us. So what's at stake? There's this concept, the ABT concept, the and of a story. You're telling a story and then this happened, and then that happened. The and, you've got momentum. You're going with the research aspects, step by step by step. And then you get to the but, that's conflict, right? And a good story's got some conflict in it, right? Good stories do. And then there's resolution. Think of your favorite movies where, there's momentum, you get to a problem, and then of course, resolution. The ABTs, this framework, this was really put together by Randy Olson, who's a filmmaker. In fact, used to be a professor and abandoned being a professor and has now been working in this I'll just say this, narrative storytelling mode, and has a really interesting book called Houston, We Have a Narrative. You know, it's like Houston, we have a problem. Houston, we have a narrative. So I would recommend that you maybe check that book out. That's sort of a really interesting resource. And so the interesting thing about and, but, and therefore is that we have a tendency when we're talking about our research to do the step by step by step by step, right? All the facts get laid out, all the facts get laid out. And sometimes the facts, I mean, we don't want to modify the facts, but sometimes they don't complete the story in a way that's necessarily interesting. And I'm going to give you an example of this a little bit later. Let's see, what is this that's shown up? I'll give you an example of that, how the order of the facts really might tell a different story or better story. You're not changing the story, but you're making the story more interesting. And then, of course, the next act, what's next in the research pursuit? You want people to be so engaged that they ask you, okay, so what are you going to do next? Okay. Now, please interrupt me if you have questions, you know, as we go along, but I want to now, let's do the first breakout. All right. And this is what I want you to do. I'm going to show you, I think in the chat, I have put a link, and this will make it easy for you to go into a breakout room and read the abstract. It's important that you read the abstract, and I want you to transform an abstract. This is an abstract that was written. There are details about or a strategy for writing science, which is a different strategy from what we're talking about today of telling a story orally, okay? So the way that you might put the story together for an oral presentation is absolutely different from what the expectation might be for submitting an abstract, as you will see, okay? Very matter of fact, but some of the elements that are necessary to pull together an oral presentation are clearly in the abstract. So I'd like you to read that. All right, so first of all, what are the basic elements of the research that's described in this abstract that you will read? And I want you to consider listing several facts from the abstract that you deem important. All right, there's a lot of detail, but read it and you figure out what parts of this abstract do you think are important for a story that you want to tell? And then I want you to write, you know, up to 5 sentences. You don't have to write 5, but no more than 5. That's going to transform that abstract as you're reading it into the framework for a story, an oral presentation. All right. And then we'll come back together and share. So this first abstract is here on the PowerPoint, but I think that Nandini has in the chat. If you go to the chat, I believe the first link is a Google Drive link, which will take you to this abstract, and you can read it perhaps a little bit. You can read it better because you're going to go into a breakout room and you won't be able to see my screen in the breakout room, but you can open the link and read this particular abstract. Okay, so I'm going to give you, we're sort of planning on about 15 minutes. And does everyone have their goal in mind? All right, you want to read the abstract, write down facts that you think are important to start telling a story about this. And then I'd like you to think about those tools that we talked about. You know, is there an opening question that you can think about? You know, what's at stake here? Is there some conflict in the story? Is there, the ands, is there a but, is there a therefore? These are the things I want you to consider when you break out into your rooms. So Nandini, are you able to?
Yes, I'm just about to open all the rooms. Can everyone access the Drive link so far? Just speak up now if you can't. You need to be logged into your.
Want to see what folks would get out of this. Because one thing it does is to let you know where you stand in terms of a scientific audience.
Yeah. All right, I think we're all back.
Okay. All right.
Hey guys, does anyone want to share what they discussed in their breakout room?
Yeah, that would be great. I'm just trying to, let's see if I can get myself back here. Okay. So we had 3 breakout rooms, right? And I'm getting, and it does not surprise me, this is not anyone's particular field that is talked about here in this abstract. But the reason I asked everyone to try to pull out of this what you thought were important facts to create a story, I'm interested to see what you pulled out and how you might start this whole discussion, or if you were giving an oral presentation. Now, one of the things that's interesting about this is, remember I said at the beginning, we're thinking about a scientific audience versus a non-scientific audience. And I said that there are obviously subcategories in each of those. So we do have non-scientists who maybe have a little bit more background in certain things, and something that they may read or be exposed to might resonate more so than obviously a real layperson with very little, if any, scientific background. Now, you all are science students and you have some background in basic concepts in biology and molecular biology. But I think you can all agree that this is very specific, detailed language here, and it in no way surprises me that you would not understand all the concepts here. And that was somewhat purposeful, to give you an abstract that was challenging. First, to see what could you pull out, and of what you pulled out, how might you then think about starting this story? So if we could have, folks from one of the breakout rooms have their discussion or talk about that with the group, that would be great. Yeah, Brooke.
So I was in group 2, and our initial discussion, like the first thing that we brought up was first the importance of the abstract and what the information that was given here. So we all came to the conclusion that like the overall basis of it was that the overexpression of the ribosomal components can lead to different diseases, which can be seen by looking at the disease's phenotypes. And so this is something that we believed would get everyone's attention because it is relevant for many different reasons.
Okay. All right, good point. Thank you. Other groups? I don't quite understand. Oh, okay, Joshua.
We tried to our main objective was to break it down into common speak, I guess is what we called it. I don't mind attempting to do that now. So basically, we tried to highlight that there was a protein from a family called ERPL22
Mhm.
that causes over that causes cells in the testes to proliferate. And we found this through using fruit flies, but we also found that this mainly happens in cyst cells and glands that use something called Gal4 drivers. So it's specific, but let's see. Yeah, we weren't able to go through all of it because it was kind of dense.
That's okay. That's all
Yeah,
right. That's
that
okay.
would be
But
like
I
the
think
start of
you
it.
have gotten, I think you have the essence. You say that this particular protein, I want to say when too much of this protein, I should say it this way, when too much of this protein is made in the testis, it has the impact of having cells divide when they should not be dividing. So that overproliferation statement is an important one. And the consequence of that is that you, I think I think many people would understand the concept of cells dividing when they should not be dividing could be a problem. Certainly could be a problem. So I think you've gotten the essence of it. I will tell you that the Gal4 story, that's a set of tricks that's useful when you're working with Drosophila or working with yeast. Gal4 happens to be a transcription factor that when it binds to DNA, it activates genes downstream. So that's sort of a tool that people that work in fly genetics can use to express genes that are sitting behind that Gal4 sequence. Okay, so if you, Joshua, based on that, did you define a question or an opening salvo for how to think about this?
Like an opening question to present to an audience.
Mm-hmm.
I think I will ask my team for assistance. Would
Okay.
Stephen, like anyone else, like to join in?
So the question I came up was, what other roles do ribosomal proteins have that contributes to the function of a cell's growth?
I like that, Jason.
Yep.
Is that right?
Yep.
I like that because the title, what does the title say? The title says multifunctional, right?
Yep.
So I think if you're thinking about a ribosome, and this is for me the exciting part, to be honest with you, about ribosomal proteins. These are components that help to make up a ribosome, and we know what ribosomes do. But if you're a component of the ribosome, and I'm telling you that now there's some other role that this protein is playing, and I'm calling it multifunctional. So that's a really key point that you've made, Jason, that you have a protein that normally we think about it functioning in a certain way. And now there's evidence that it can do something else, right?
Yes, ma'am.
Yeah, so that's a really key part of what's being presented here. Is there someone else that has another way of, or some additional information that they want to contribute to a way of thinking about presenting this, starting the oral presentation?
I was in breakout room 1,
Okay, yes.
and we were trying to focus more on like telling a story. So we thought of starting with like describing how fruit flies like don't really seem very similar to humans, but they're an important model system.
Mhm.
And they were using the ribosome in this study, and it could be related to humans. And we also thought of focusing on more of how the mechanism is unknown. So kind of using that as suspense in the presentation, and then
Okay.
ending with how there's more experiments that are ongoing to discover more about this problem.
Okay, I like that. I like that too. That's great to have taken. So you've connected fruit flies, which there are some politicians that don't understand that fruit flies are really saviors in many ways. Not too many people get upset about sacrificing fruit flies, but they have a lot of the same genes that we do, and we've learned a lot. So I like that approach to grab the attention of someone with that, and then to go from there. I think that's great. And the mechanism, again, unknown, what's happening here. So that's the intrigue. And then sure, there's a lot more to be done. Yeah, thank you, Penny. Any other comments? Okay, so I want to make sure I'm going to move to I want to reinforce the idea that many of you have slightly different backgrounds, right? That would allow you to pick up different messages from this abstract. And I think we have evidence of that. If I were telling this story, can everyone see my screen at this point? Are you?
Yeah.
Okay.
You can see it.
So I'm going to just quickly tell you a little anecdote that I use to talk about multifunctional proteins. And I think I may have shared this anecdote with some of you at some point in time. Many, many years ago, I took ballroom dancing at Lehigh with a faculty colleague. You don't know this colleague because he has retired before any of you actually, I believe, would have come to Lehigh. But at any rate, we had this idea. They offered ballroom dancing, and my colleague and I said, hey, let's do ballroom dancing. Okay. So we went to Taylor Gym, and they would have classes, and we were learning, the tango and waltzing and all the classic ballroom dances. And it turns out that my partner could do ballroom dancing with me as the waltz. I mean, we could waltz together and carry it out and not look like complete clowns. However, when it came to the tango, he was miserable. It was so bad. And so I needed to learn the tango. So there was we would switch partners. We would essentially switch partners. And I had another partner that I could do the tango with, but I could not do the tango with Jeff Sands. That was just not happening. And believe it or not, when I started studying this family of ribosomal proteins that have different functions, I thought of ballroom dancing with Jeff Sands and the other person. So when Jeff Sands and I could dance together, we could do the waltz, but I had to switch partners in order to do the tango. All right. So if I were telling this story about multifunctional proteins to, a scientific audience or even a non-scientific audience in particular, especially a non-scientific audience, I could start this whole story, this whole presentation with ballroom dancing and switching partners, right? Switching partners allows us to do a different task, a different type of dance. And so at the same time that I'm talking about ballroom dancing, I'm also getting a message, hopefully, to stick in the minds of the audience. So in other words, the take-home message here is that, and some proteins can carry out different functions, obviously, depending on what proteins they bind to. If you bind to protein A, all right, if I'm dancing with Jeff Sands, protein A, we can do the tango, pathway A. But if bound to protein B, that's the other person, we can then function in a totally different pathway. I can do the tango, right? So multifunctional proteins carry out different functions depending on their binding partners. But this is key. But if we disrupt the binding of a partner, that can have consequences. Therefore, remember the ABT here. Therefore, disease can be caused when normal pathways are not working. So if I wanted to get the take-home message to people in this way, I could use that anecdote to start my story about this multifunctional protein research that we're doing in the laboratory. Any comments about that? Did you guys get it? Does it make sense?
I think it's a really great analogy. I have no idea how that came to you, but it makes so much more sense now that you explained it that way.
Yeah, well, I have to just say that, I mean, it was a real experience for sure. And when I think of, you know, binding and partnering, it really literally, I thought back to those days. It's like 20, 25 years ago, doing ballroom dancing with Professor Jeff Sands. So yeah, that helps me to explain a number of things. So if you want to remember, the essence of this abstract is really about this, right? And going into a different pathway in this sense is that now we have one of the protein partners, the L22, goes into a different pathway of stimulating cells to grow when they should not be, over proliferating. And so again, that's a take-home message. All right, we have only a few more minutes, and this is what I'm going to change this up a little bit and not necessarily have you go to breakout rooms, but I'm going to tell you a story. I want you to, this is complicated, and I think there are a couple people here in the session that have a sense of what's going on here. I know Penny is one of them, but I'm going to tell you this story, give you the essence of this abstract in my own words without your having necessarily to read the whole thing. And this is related to the phage work that goes on in my lab. And so one of the things that we're interested in the phage story is how phage genomes evolve. Phages have lots of genes for which we don't know what those genes do. And in fact, the reason that we would be interested in this is because, to be honest with you, bacteria can be our friend and they can be our enemy. And we tend to think of bacteria from an enemy point of view, especially when they are resistant to antibiotics. And so the question becomes, when you have a pathogen, what can you do to eradicate a pathogen? And phages, which are, you know, bacterial killers, are really a possibility. And this whole notion of phage therapy is something that we're interested in. But we are interested generally in how phage genomes are organized and what are the genes. So this abstract is not necessarily related to phage therapy, but it does have an impact on our understanding of how we could use phage therapy. All right, so I want to tell you that this abstract is related to infection of a particular phage in a bacterium that's harboring another phage, all right? So Butters is hanging out in this bacterial cell. It's not killing it. It's just hanging out, living with the bacterial cell in such a way that it protects the host, the bacterial cell, from infection by other types of viruses, other types of phages. And one of those other types of phages is called Island 3. All right, it's this different name, Butters Island 3. So it turns out that Butters is mounting a defense. If Island 3 comes along, Butters expresses some genes that prevent Island 3 from killing the host. Now we're interested in what those genes are. And so that's a strategy, that's a rationale for trying to understand basic things that happen between phages and host cells to provide some protection. Okay, so it turns out that when Island 3, as I said, infects a bacterium that harbors Butters, every now and then there are Island 3 phage that can break through the defense. You know, it's like breaking down a barrier, you know, knocking down a door in some way. And if you have an opportunity to figure out what that is, it turns out that you could learn something if you can isolate phages, Island 3 phages that somehow could override the defense. That'll allow you to learn something about what the defense is. So that was our goal originally in this set of experiments. And what we found out is that instead of getting defense escape mutants of Island 3, ones that had broken down the barriers to defense, we got instead hybrid phages. So those Island 3 phages were now recombining their genomes with the Butters genome. And so what you get out of that infection is not either Butters, it's not Island 3, but it's a Butters Island 3 hybrid. And that's what this whole abstract is about, okay? Now, the way the abstract is written, it's based on the recombination events. But I want to tell you that the way in which we uncovered all this was the order in which we uncovered all this is not the order in which I told you. But it makes a better story if I tell you from the beginning of this that, you know, if I were to ask you to pick out important points from this particular abstract, you would at some point in time see that this line that says we have previously reported, that's a key that something happened early in this investigation. But you see in the abstract, it really comes like a third of the way into the abstract. But that's actually the first part of this story. So if I were telling this story the way that it really has happened, I would start with our intentions of isolating mutants, but that didn't happen. And we then learned something about this whole process of creating hybrid phages. So the very last for fun thing, and I know we maybe can take 2 minutes for this. I want to ask, has anyone seen the movie Shang-Chi and the Legend of the Ten Rings? Anybody seen that film yet? No? I don't want to be a spoiler.
Not yet. I want to watch it
Not yet?
soon though.
Okay, all right. So I'm not going to give it away because I want to remember part of the oral presentation is that you want people to think, oh, what's next? What's next, right? So I want you, if you get to see The Legend of the Ten Rings, all right, I want you to think about two phages battling each other, all right, defending each other. I want you to be thinking about this connection and how it might relate to the story that I told you. So, you know, I'm leaving you something to think about. And then the last thing here, it says, when is the sequence 3, 1, 2, 4, 5 better than 1, 2, 3, 4, 5? All right, so what I was going to have you do from the other abstract was to, again, write down 5 facts that you pull out of the abstract in the order in which you pulled them out of the abstract. But it turns out that if you put fact number 3 before facts 1 and 2, you tell a better story. All right, if you actually let people know that we were really trying to isolate defense escape mutants, and we stumbled upon this, right? This is the conflict. This is the thing that makes it an interesting story. Sometimes you have to reorder the sequence of events in your research to tell a story that's more compelling. And with that, I'm going to end and ask if you have any burning questions, burning comments to make.